compound 3-keto-24 epi 28 nor catasterone. In Chlorella vulgaris, on this algal model, I released many studies. It's a member of Treboxophyta class. Multiple brassinosteroids have been identified, including brassinolide, castasterone, and 6-deoxocastasterone. This species shown a particularly high concentration of brassinosteroids. Additionally, the Chlorophyta and Treboxophyta class trend to have a higher concentration of brassinosteroids compared to other algal groups. For example, in Chlorella vulgaris, we observed brassinolide at 70 picogram per gram fresh weight and castasterone at 470 picogram per gram fresh weight. Additionally, other brassinosteroids such as 6-deoxocastasterone, 6-deoxotifasterol, 6-deoxoteasterone are present in this species. On the other hand, in Acutodesmus obligus, we see lower concentration of brassinosteroid measured at 1.41 picogram per gram fresh weight and 28 homobrassinolide and only small concentration. This concentration, however, shows significant variability across algal species, which can depend on both the species itself and the environmental conditions they were exposed to. Brassinosteroids belong to a class of steroidal triterpenes, which are molecules made up to 27, 28, or 29 carbon atoms. The structure is composed of 3 terpene units or 6 isoprene units, which form the core structure. The brassinosteroids can be categorized into different skeletal structure types. For C27 brassinosteroids, such as those derived from cholesterol, for C29 brassinosteroid derived from campesterol, and for 29 brassinosteroid derived from beta-cytosterol. Each of these variations in structure plays a role in the activity of brassinosteroids in plants and especially in algae. The core structures can also vary based on specific chemical modification, such as 7-oxalactone, 6-hydroxy, 6-oxo, or 6-deoxy group found in different brassinosteroids, like brassinolide, tiasterone, castasterone, and 6-deoxocastasterone. This slide presents the structure of brassinosteroids, only compounds which were detected in algae. The red color means that's C27, the blue color is C28, and only 2 compounds belong to C29 brassinosteroids. Of course, this is a total number of carbon atoms in molecules. Only one from C27. It's a 3-keto-2022 epi 28 norcatasterone occurs. Environmental factors such as salinity, stress, and exposure to heavy metals can significantly increase the levels of brassinosteroid in central algal species like Spirulina platensis. This stressor trigger the production of brassinosteroids, which help the alga cope with challenging environmental conditions. However, detecting brassinosteroid can be quite challenging due to the low concentration in algae. The methods used to detect these compounds are source-intensive, involving complex techniques such as LC-MS and or GCMS. There are, of course, also some gaps in research, mainly due to the high cost and limited availability of algal samples, which makes large-scale studies on the occurrence of brassinosteroid more difficult. Nevertheless, overcoming this challenging is crucial to advancing our understanding of brassinosteroid and their role in algae. As I mentioned earlier on the previous lectures, too, the low concentration of algae in low concentration in algae brassinosteroids presents a significant challenge for detection. One of the most common methods which I recommended to you is liquid chromatography mass spectrometry. These techniques allows the high sensitivity and precision, separating compounds before analyzing them with mass spectrometry. Another, of course, which I used to many years ago, was gas chromatography mass spectrometry, GCMS, which is suitable for volatile derivatives of brassinosteroids. However, because brassinosteroids are not inherently volatile, they require derivatization before analysis, adding an extra layer of complexity. Despite these challenges, both LCMS or GCMS are crucial tools in the study of brassinosteroid, enabling us to quantify and understanding their presence in various algae species, of course. One promising method involves the use of enhanced LC-MS techniques, which can increase sensitivity up to close 8,500 times, especially using isotopic labeling such as deuterium or tritium. This allows for precise quantification of brassinosteroid even at very low concentrations. However, one limitation of isotopic labeling is the possibility of isotopic exchange during the analysis, which can affect the accuracy of results. Another useful method is immunochemical assay. I didn't recommend this, but it is more cost-effective and suitable for real-time analysis. These assays use only some monoclonal or polyclonal antibodies to detect brassinosteroids, but their specificity can be lower due to the potential cross-reactions with other substances. Moreover, they may not be as sensitive with dealing with very low concentrations of brassinosteroids. While these techniques are effective, there is still room for further optimization to improve sensitivity and reduce limitation in brassinosteroids detection. Now let's take a look at some emerging techniques in brassinosteroids. They are not very popular. One such method is proteomic analysis, especially two-dimensional difference gel electrophoresis. The techniques can be used to identify proteins that are regulated by brassinosteroids. It's not, of course, techniques to detect a brassinosteroid and check the presence, their concentration. Its approach provides deeper insight into the cellular processes influenced by brassinosteroids. Another promising technique is isobaric tagging of for relative and absolute quantification, which labels peptide for both relative and absolute quantification. Why this method offers great potential, they do come with some challenges. And for example, the data analysis process can be quite complex, and there is variability in plant proteome that could affect the results. Let's now summarize the key insight from emerging techniques in brassinosteroid detection. CMS remains the most commonly used methods for detecting brassinosteroid in algae. This method have proven to be effective, but emerging techniques like proteins, which I told 2 minutes ago, show promise to providing deeper insight into brassinosteroid regulated proteins. It's very important, of course. Looking ahead, there are several future detections in brassinosteroid detection. One of the goals is to develop more cost-effective and sensitive methods for detecting brassinosteroids, especially particularly at low concentration. There is also the potential for integrating proteomic analysis with other detection techniques to create more comprehensive understanding of brassinosteroid function. The biosynthesis pathway of brassinosteroids begins with isoprene units, which form squalene and cycloartenol. These compounds then diverge into two main oxidation pathways: the early C22 oxidation pathway and the late C6 oxidation pathways. Both of these pathways ultimately lead to the formation of brassinosteroid and the most active brassinosteroid and the last compound in the pathways. Brassinolide is the most well-known brassinosteroids, not only present in plants, algae, and applied to studies. Similar to 24-epibrassinolide, in algae there is an interesting distinction. The precursor of for brassinosteroid biosynthesis are produced via the non-mevalonate pathways in plastids, which is different from higher plants that synthesize from precursors in the cytoplasm. This unique feature of algal brassinosteroid biosynthesis is one of the key areas of study, as it could provide insight into the evolution of brassinosteroid pathways in different organisms. Among the important intermediates are compounds such as campesterol, campestanol, catasterone, and 6-deoxycastasterone, which are all precursors in the formation of brassinosteroids. There are also several critical enzymes that play a key role in the biosynthesis process. For example, it's DET2 reduce ergo-4-3N to ergostan-4-1. And a crucial step in the pathway. The next is dwarf 4 hydrolyze brassinosteroid precursors at the position C22 and BR6OX1 enzyme adds a ketone group at the sixth position carbon. This enzyme regulates the formation of different types of brassinosteroids. There are totally 70, and ensure the proper production of compounds like brassinolide, which are of course essential for algae and plant development. This slide presents the brassinosteroids, the biosynthesis of brassinosteroids in higher plants and algae, where the name in the boxes indicate detection in both higher plants and algae, with a particular emphasis on algae. You can see on the end, I will show some references, and I recommend one paper where is this scheme present. As you can see, only one of the three pathways has been fully discovered, similar, of course, to the higher plants. The rest, C29 brassinosteroid pathways, is only partially understood, while the C27 brassinosteroid pathway is yet not to be discovered. It's a chance for us, it's a chance for you, of course. Brassinosteroid biosynthesis is heavily influenced by environmental stressors such as salinity and exposure to heavy metals. These stressors can trigger a change in biosynthetic pathways, causing algae to produce higher levels of brassinosteroids as a stress response. One of the fascinating aspects of brassinosteroids biosynthesis in algae is its pathway flexibility, which allows the organism to switch between different oxidation routes depending on the environmental conditions. Of course, more research is needed to uncover the specific enzyme activities and genetic regulation involved in these processes. It's not fully understood in algae, but vascular plants is. Brassinosteroids play a vital role in stimulating cell division and elongation process. They are crucial for the overall growth of algae. They also enhance the biosynthesis of key cellular components such as proteins, nucleic acids, saccharides, lipids, and all of which are necessary for healthy cell function and growth. In addition to supporting growth, brassinosteroids improve the net photosynthesis rate. Professor Shamsul Hayat present these reactions on his lectures, and brassinosteroid increase chlorophyll contents, both of which of course are essential for energy production in algae. Furthermore, brassinosteroid influence physiological responses by increasing cellular adaptation to stress and regulating carbohydrate assimilation and allocation within the cells. This function highlight the importance of brassinosteroids not only in promoting growth but also in the helping algae to adapt and thrive under challenging environmental conditions. Brassinosteroid boosts photosynthesis by increasing the level of chlorophyll A, chlorophyll B. This enhancement improves the energy efficiency in algae. Brassinosteroid also stimulate the production of carotenoids, especially lutein of astaxanthin, which is important, for example, in Hematococcus pluvialis. They also enhance the production of protein and extracellular saccharides, glucose, fructose, and others. Brassinosteroid also increase its phenomenon, the secretion. It's extracellular secretion of sugars and other organic compounds, for example, protein, which can be used by biofuel production and of course interact with other organisms in aquatic environment. For optimal metabolic activity, brassinosteroids are most effective at the concentration which is present. This range of concentration has been found to significantly stimulate metabolic activity and enhance the production of these key metabolites. Under abiotic stress conditions such as salinity and temperature extremes, brassinosteroids help reduce the accumulation of reactive oxygen species in the cells. We know about reactive oxygen species that they can cause oxidative damage, and brassinosteroids play a crucial role in protecting algal from this damage, similar in other plants, activating enzymatic oxidants and non-enzymatic antioxidants like glutathione, ascorbic acid, and other compounds. We know that this activation of antioxidant enzymes works synergistically with other phytohormones, such as auxin and cytokinins, to enhance the algal overstress resilience. The role of stress markers, not only in algae but also in plants, I presented earlier, so I omitted this knowledge. The combined action of brassinosteroids and other phytohormones have algae to be for cope with environmental stresses, making, of course, brassinosteroids essential for maintaining cell integrity and function under challenging conditions. One of the most significant effects of brassinosteroids is their ability to increase cell division and the size. Chlora vulgaris has a special big giant cell, which divided normally from one mother cell into 4 daughter cells, but after brassinosteroid treatment, from one mother cells divided into 16 daughter cells. It's a really look fine when you can see biggest cells. It's a giant cell. It's fascinating. For example, of course, I provided in the studies in Chlorella vulgaris the application of brassinosteroid resulting doubling cell and size. The other key benefits of brassinosteroids is their role in lipid accumulation. These hormones manipulate lipid profiles, making them highly valuable for biofuels production and pharmaceutical application. One specific brassinosteroid, 24-epibrasinolide, has been shown to enhance both minor and major fatty acid production, which is crucial for optimizing lipid contents in algae. We know that Chlorella vulgaris is very popular as in medicine shops and as a supplement of our diet and in tubs or as a powder. Additionally, different brassinosteroid can be treated for specific metabolic outcomes. Compounds such as brasinolide or 24-epibrasinolide exhibit distinct effects on algal metabolism, showing the promising control over biomass production. What about interactions of brassinosteroid in other phytohormones? The synergistic interactions between brassinosteroids and, for example, auxin, cytokinins, gibberellin, abscisic acid exist. One of the most significant interactions occur with auxins, particularly in the oleophilic acid. Brassinosteroids stimulate the expression of PIN genes, which are crucial for, as we know, for auxin transport. In turn, auxins enhance the endogenous level of brassinosteroid in algae is very fascinating and very important. And very stimulating to our next future studies on a molecular, of course, level, not metabolic or molecular level. Together, these two hormones promote increase protein, sugar, and pigments production. That's mean, of course, chlorophyll, carotenoid, xanthophylls, carotenes, alpha, beta carotenes. Another important interaction is with cytokinins such as transxanthines. When brassinosteroid and cytokinins work together, they enhance cell proliferation and improve metabolic outputs. This includes increase, of course, similar chlorophyll biosynthesis and enhance saccharide production, both of which are vital for energy metabolism in algae. Additionally, brassinosteroids play a role in biosynthesis of abscisic acids, includes ABA production, which helps algae become more resilient to drought and salinity stress. This collaborative effect strengthens the ability of algae to survive in challenging environmental. These interactions show that brassinosteroids do not act alone, but rather function as a part of network complex, hormonal network complex that regulate algal growth, metabolism, and stress adaptation. Brassinosteroids also enhance the production of carotenoids, such as astaxanthin, especially in Haematococcus pluviale. Is algae very useful in applications when acting synergy with yasmonic acid and salicylic acid. This interaction is particularly important for algae. Use in the production of pigments and astaxanthin for industrial and pharmaceuticals applications. Brassinosteroids regulate and improve antioxidant response, providing protection against abiotic stress such as extreme temperature and oxidative stress. These protection effects ensure better survival and physiological performance of algae under unfavorable conditions. Another crucial aspect of brassinosteroid is ability to regulate of course, biosynthesis, influence of auxin and cytokinase production, and other, of course, processes. What about the role of brassinosteroid in stress responses? Brassinosteroid mitigate effects of abiotic stresses. As you see, salinity, temperature, extreme low and higher temperatures, and heavy metal toxicity. Brassinosteroid, of course, act by reducing reactive oxygen species accumulation, and the key benefits is that protect cellular structures for oxidative damage and just stress tolerance. I told you about that earlier, that brassinosteroid increase activity of catalase superoxide dismutase ascorbate peroxidase, of course reduce lipid oxidation, and maintains membrane integrity. The impact of on reactive oxygen species is lowers hydrogen peroxide and superoxide ion levels, and support, of course, cellular survival under these stresses. And how brassinosteroid can help algae cope with salinity and temperature stresses. In the case of salinity stresses, brassinosteroid help stabilize ion contents within the algal cells, preventing osmotic imbalance. They also boost the production of antioxidant compounds, which cut down on the harmful effects of reactant oxygen species generated by high salt concentration. This production enables algae to maintain their growth and the metabolic function even in salinity stress. Under temperature stress, whether due to the extreme heat or cold, brassinosteroid help preserve chlorophyll levels and maintain carbohydrate metabolism. This ensures that algae can continue photosynthesis efficiency despite fluctuation in temperatures. Additionally, what is maybe new brassinosteroid enhance the production of abscisic acid, a hormone that play a key role in stress adaptation, further improving the algae resilience. What about heavy metals in the environment? We know that can be toxic to algae. Heavy metals can be toxic to algae, leading the oxidative damage, disturbing metabolism, and reduced growth. However, brassinosteroid play a role in mitigating these harmful effects. One of the primary metabolism is detection and protection. We know that brassinosteroids stimulate production of phytohormones to detoxify heavy metals. Phytohormones are molecules that bind to heavy metals ion and neutralize their toxicity. Earlier I present the biosynthesis of phytohormones, so you know about it. This prevents heavy metals from accumulating at dangerous level inside algal cells. Additionally, brassinosteroid enhance antioxidant enzyme activity, which help counteract the oxidative stress caused by heavy metal exposure. Enzymes are the same, of course, catalase, superoxide dismutase, ascorbate peroxidase, which protect, of course, the cellular structures. As a result, algae treated with brassinosteroid maintain higher photosynthetic efficiency and overall growth even in polluted by heavy metals environment existed. This capability makes algae a promising candidate for phycoremediation and bioremediation, where they can be used clean up heavy metals polluted water through natural absorbing and detoxification processes. The graph summarizes the application of brassinosteroid in algae, showing anti-stress function. This graph comes from a publication which I published together with Professor Shamsul Hayat some years ago. The future research direction in brassinosteroid studies, particularly of course in algae, should focus on elucidating the C27 and brassinosteroid pathways. Of course, on 28 brassinosteroid pathways, it's not fully complete, but also two. Investigation that biosynthesis and functional role is, we know that remain undiscovered in algal cells. What else? Comparative analysis with higher plants, understanding the similarities and differences in brassinosteroid pathways and function between algae and higher plants, physiological and ecological function. We should explore the role brassinosteroids in algal growth, stress tolerance, and adaptation to aquatic environments. And then regulatory mechanism. It's still remain unclear. Identifying the molecular signaling pathways and the receptor mechanism of brassinosteroids in algae is very, very important. We don't know about anything of brassinosteroid signal transduction in algae. And the last biotechnological application, investigation how brassinosteroids can be used to enhance algal biomass production, biofuel generation, and stress resistance. And what else? Evolutionary perspective, omics-based approaches. Utilizing genomics, transcriptomics, and metabolomics to uncover new brassinosteroid-related genes and pathways in algae on, for example, enzyme activity, genetics regulation are needed to clarify they may be or pathways biosynthesis mechanism. We start this study on genomics. Finally, integrating modern technologies into brassinosteroids research could revolutionize the field of plant hormone. Genetic engineering could be used to enhance endogenous brassinosteroids in algae, leading to improved growth and resilience. Moreover, developing cost-effective methods for large-scale brassinosteroid production in algal cultivation could unlock new opportunities for biotechnology and industry. I told that brassinosteroid enhance lipid production for biofuel industry, nutraceuticals, pharmaceuticals, and wastewater management, which is very important. Another benefits, of course, that benefits. Yeah, we have seen that how brassinosteroids play a crucial role. Phytohormones regulate fundamental processes like cell division, photosynthesis, and other. On the end, I would like to present the last summary of brassinosteroid occurrence and function in algae. I would like to recommend article with my PhD student on the same title around the brassinosteroids in algae, which was published in Algal Research. Oh, this is the references. The last Żeruń, Gian, Bajguz, Andrzej around the brassinosteroid in algae, Algal Research, 2025. And the next, of course, chapters published in brassinosteroid plant growth and development, edited by Shamsul Hayat, Mohamed Yusuf, Renu Bardwai, and Andrzej Bajguz. It was in 2019, 6 years ago. Yeah. Thank you very much.
another newly discovered plant hormone, hydrogen peroxide. In my preceding lecture, I had discussed detail the role of polyamines in abiotic stresses in plants. So today I will be just navigating specific roles of hydrogen peroxide in plants under abiotic stress. So this is a very fitting topic to the Gian program. So I will discuss some of the case studies done from my lab. So in my previous lecture, I would like to take up with the last keyword of my topic, that is role of hydrogen peroxide in plants under abiotic stresses. So the last keyword is stress. So in my previous lecture, I had discussed what stress is basically, and as you know that these stresses are of two types, abiotic and biotic stresses, and these in both biotic and abiotic stresses challenge the ability of the plant to perform well in adverse environmental conditions. So you see that plants generally exposed with wide range of abiotic stresses and these abiotic stresses generally affect growth, productivity, and these abiotic stresses, as we are discussing from the last 3 days, includes mainly drought, salinity, extreme temperatures, heavy metal toxicity, and nutrient deficiencies. And all these stresses basically disrupt physiological and metabolic process of the plants. But out of these stresses, particularly drought and salinity, are very detrimental and reduce photosynthesis and water use efficiency. So on the other hand, if you see the biotic stresses results from the interaction with the living organisms such as pathogens, insects, nematodes, and weeds. So generally these stresses generally compromise the immunity of the plant, leading to loss of yield. And the combined effect of biotic and abiotic stress, just I mentioned that it could be interactive, synergistic, and making it very fatal to the plant survival. So sometimes what happened that both plant face biotic as well as abiotic stress. So the combined effect could be additive, synergistic, making it very fatal to the survival of the plant. Now generally, as you see in this slide, that plant, they are exposed to a number of abiotic stresses like heat, chilling, drought, waterlogging, salinity, and plants generally cope with these stresses by modulating activity of antioxidant enzymes, and these stresses also disrupt the activity of functioning of photosystem first, and second alter the activities of photosynthetic enzymes, reduces the stomatal conductance, and trigger loss of water. In addition to these stresses also cause hormonal imbalance. So this is also very pertinent that these stresses generally cause hormonal imbalance. Now I will just give you a brief overview how reactive oxygen species are produced in different cell organelles. Just I little bit elaborated in my previous lectures. So because also this hydrogen peroxide is very important as one of the antioxidant, so I will discuss it a little bit in more elaborate way. So you see that reactive oxygen species generally they are central to the plant responses to abiotic stresses. So what is the fate of this reactive oxygen species under normal conditions? So basically these are produced in a small amount as a byproduct of metabolic processes, and they play a very critical roles as a signaling molecule in growth and development. This is the fate under normal condition, but what happens during abiotic stresses? Excessive production of reactive oxygen species disrupts cellular redox homeostasis, leading to oxidative stress that damages lipids, proteins, and nucleic acids. So you see, in one way it's produced as a normal byproduct and imparting signaling functions to regulate growth and development, but excessive production of reactive oxygen species leads to damage of lipids, protein, and nucleic acids. So how this reactive oxygen species is produced in chloroplast? So generally during abiotic stresses like high light intensity or during drought stress, there is an increased production of reactive oxygen species in chloroplast, and you see that excessive light energy generate generally singlet oxygen species and superoxide radicals in photosystem first and second, and there is a one important enzyme, superoxide dismutase, generally converts superoxide radical into hydrogen peroxide, which acts both as a signaling molecule and as a potential potential oxidant. So you see that hydrogen peroxide is playing a dual role. In one way it is acting as a signaling molecule, in other way it is acting as a oxidant. So what is the fate of these reactive oxygen species production in mitochondria? So generally in response to abiotic stresses such as salinity, high temperature, generally ROs are produced in the mitochondrial electron transport chain, and mitochondrial production of reactive oxygen species, they are generally closely linked to energy metabolism and stress signaling pathways. What is the fate of these reactive oxygen species in peroxisomes? So during abiotic stresses like drought and high light, photorespiration in peroxisome is generally intensified, leading to increased production of hydrogen peroxide by enzyme glycolate oxidase. So peroxisomal production of reactive oxygen species are very critical for the metabolic adjustment during stress. So among the various reactive oxygen species, hydrogen peroxide basically plays a very central role due to its ability to diffuse across membrane. So this is very important point, you must focus on this, that hydrogen peroxide plays a central role due to its stability and ability to diffuse across membrane. Other molecules cannot easily diffuse the plasma membrane, but hydrogen peroxide can diffuse plasma membrane. So in this respect, hydrogen peroxide is very important as a signaling molecule, and you see that hydrogen peroxide acts as a double-edged sword. It serves as a, in one way it serves as a signaling molecule to activate defense mechanism, but can also cause oxidative damage if its concentration is not regulated. So this dual role of hydrogen peroxide makes a key mediator in plant responses to abiotic stresses. So by understanding this sort of connection, we can explore strategies to balance hydrogen peroxide levels for improved stress tolerance in plants. So you see that concentration of hydrogen peroxide is very crucial. In one way it's acting as a signaling molecule, in other way it's acting as a oxidant. So it's acting as a double-edged sword. Now hydrogen peroxide serves as a, generally a delicate balance is maintained between beneficial signaling and harmful oxidative stress in plants. So you see that from this slide you can have a view that there must be a balance should be maintained between its beneficial signaling and oxidative stress. So generally, as I mentioned, that at low concentration it acts as a signaling molecule regulating processes like stomatal closure, gene expression, activation of antioxidant defense system. However, excessive accumulation of hydrogen peroxide, often due to abiotic stresses, leads to oxidative damage to lipids, protein and DNA. So, this dual nature of hydrogen peroxide makes it both as a protector and as well as a threat, depending upon its level and ability of the plants to maintain a redox homeostasis through efficient antioxidant system. So, how hydrogen peroxide in plant is produced through both enzymatic and non-enzymatic pathways, and as I mentioned earlier also that enzymatic sources include NADPH oxidase on the plasma membrane, which generate superoxide radicals that are converted to hydrogen peroxide by superoxide dismutase. Additionally, just I mentioned in my preceding slides that peroxisomes also contribute significantly via beta oxidation of fatty acid and photorespiration, where glycolate oxidase is a key enzyme responsible for this. Chloroplast and mitochondria also produce hydrogen peroxide as a byproduct of electron transport chain. In addition to this, non-enzymatic sources of hydrogen peroxide include spontaneous reactions such as dismutation of superoxide radicals in the apoplast, and the controlled generation and localization of hydrogen peroxide are very essential as it plays a dual role in signaling and oxidative stress depending upon its level and cellular environment. This makes hydrogen peroxide a versatile molecule in plant physiology and metabolism. Now, how hydrogen peroxide is detected by the plants, how it is perceived on the cell membrane. So, generally proteins present on the plasma membrane can detect reactive oxygen species like hydrogen peroxide and acts as a signaling intermediate by directly interacting with hydrogen peroxide. Although you see that hydrogen peroxide is very small, too small to have a specific receptors, its oxidizing nature allows it to modify thiol residue present in the proteins, and extracellular hydrogen peroxide generated in the apoplast by enzymes like NADPH oxidases and superoxide dismutase. They are generally induced by various abiotic stresses which enters into the cytosol through aquaporins. Once hydrogen peroxide reaches inside, it oxidizes cytoplasmic proteins to regulate stress responses and growth. Recently, a major breakthrough in hydrogen peroxide signaling was achieved by identification of HPCA1, which is a leucine-rich repeat receptor kinase located in the plasma membrane. And this HPCA1 is generally oxidized by hydrogen peroxide on the thiol group of cysteine residues in its extracellular domain. So, this is a major breakthrough achieved in the signaling of hydrogen peroxide, and this oxidation basically activates autophosphorylation of HPCA1, which in turn triggers calcium channels and induces a stomatal closure. This discovery basically signifies and highlights the role of HPCA1 as a critical component of hydrogen peroxide perception and stress adaptation, offering a new insight into cell surface ROS sensing mechanism. Now, how hydrogen peroxide is transported? So, hydrogen peroxide transport across membrane is a very critical aspect of its role in signaling, but its polar nature poses a challenge for diffusion through hydrophobic membranes. To overcome this problem, hydrophobic nature of hydrogen peroxide, you see that hydrogen peroxide is transported via aquaporine channels which facilitate its movement to regulate protein activity and signaling in the cytoplasm. And aquaporins homologs specifically from TIP and PIP families play vital role in selecting transport of hydrogen peroxide. In addition to these, a unique model of hydrogen peroxide transport in chloroplast have been proposed. I will discuss that. You see that the level of the effect of hydrogen peroxide varies significantly, with its level ranging from beneficial as a signaling molecule at lower concentration, triggering a programmed cell death at higher concentration. So what happens at higher concentration of hydrogen peroxide? Generally oxidative stress is stimulated. So excessive presence of hydrogen peroxide leads to oxidative damage by inducing antioxidant system, causing lipid peroxidation, protein carbonylation, and DNA damage. Excessive concentration of hydrogen peroxide also damage DNA and repair. Generally, hydrogen peroxide induces DNA strands break and base modification. So this is a very crucial change brought about by hydrogen peroxide in DNA. If you see that repair mechanisms are insufficient, it can trigger cell death pathway. Now, programmed cell death is also one of the important repercussion of higher concentration of hydrogen peroxide. That high level of hydrogen peroxide levels basically activate the mitochondrial pathway of programmed cell death. So what happens at lower concentration of the fate I described, what happens at higher concentration of hydrogen peroxide. Now I will discuss little bit what happens at lower concentration of hydrogen peroxide. Generally it acts at a lower concentration, it acts as a signaling molecule and serves as a secondary messenger activating redox sensing proteins and signaling pathway. It modifies cysteine residues in redox sensors like thyroidoxin and glutaredoxin, leading to changes in their activity. Then at low concentration it also alters gene expression. Activation of various transcription factors are well studied. It leads to closure of stomata. Basically hydrogen peroxide signals guard cells to close stomata, reducing water loss during drought or high temperature. Basically this involves sort of ion channel changes in ion channels and calcium signaling. Lower concentration of hydrogen peroxide is also very much crucial for cell wall reinforcement and also lipid peroxidation. Now, what is the role of hydrogen peroxide in photosynthesis? Just nutshell I would like to discuss. Basically, hydrogen peroxide at lower concentration protects photosynthetic apparatus of the plants during stress. It also regulates stomatal opening and optimizing uptake of carbon dioxide. In addition to this, you see that it mitigates, I am talking about the low concentration. So low concentration of hydrogen peroxide basically mitigates photo inhibition under high light intensity. It also enhances repair mechanism for damaged proteins, promotes electron flow balance in photosystem 2, first and second. Now you see this table reveals basically the impact of hydrogen peroxide on various photosynthetic traits in different crop plants like Zea maize, Vigna areata, Oryza sativa and tomato crops. So if you want to have a quick look of the work done in relation to hydrogen peroxide, you can have from this slide. Now what is the role of hydrogen peroxide in general in stress tolerance? So generally a rise in hydrogen peroxide levels a rise in hydrogen peroxide levels triggers several cellular responses, including modulation of intracellular calcium levels, which is very critical for activating protein kinase that initiate stress-related signaling pathways. So as a signaling molecule, you see that hydrogen peroxide influences the activity of transcription factors in the nucleus. So to counteract the potential harmful effect of excessive ROS production, basically plants have developed a hydrogen peroxide scavenging system that includes antioxidant enzymes like catalase, ascorbate peroxidase, as well as non-enzymatic antioxidants such as ascorbate and glutathione. So these antioxidants basically works in concert to detoxify hydrogen peroxide and maintain cellular redox balance, preventing oxidative damage to lipids, protein and DNA. So these are basically if there is any fine tuning in the regulation of hydrogen peroxide and its scavenging, the plant's ability to cope with the stress will enhance, promoting resilience and survival under adverse environmental conditions. So you see from this table describe the effect of hydrogen peroxide mediated abiotic stress tolerance in different crop plants. So as a reference, you can have a look of this table. Now, what is the role of hydrogen peroxide in drought stress? So you see that hydrogen peroxide basically plays a very critical role in plant responses to under drought stress by acting as a signaling molecule that mediates several adaptive processes. It induces stomatal closure, reducing water loss through transpiration. Basically, what hydrogen peroxide does basically, it activates drought responsive gene pathway, enhancing its production of protective metabolites that helps to mitigate cellular damage. Additionally, hydrogen peroxide also regulates antioxidant enzyme activities, maintaining a redox homeostasis under stress condition. Furthermore, hydrogen peroxide interacts with also one of the important growth hormone, that is abscisic acid, which is responsible in drought signaling to coordinate responses of plants that optimize water conservation and cellular protection. Now you see this figure basically demonstrate that S2 treatment could help to mitigate drought-induced morphological damage in soybean plants. So plants under severe drought condition with application of hydrogen peroxide show better stress tolerance compared to untreated severe drought stress plants, highlighting the protective role of hydrogen peroxide in drought stress. So you see that this figure shows the effect of drought stress on soybean plants on various physiological parameters with or without application of hydrogen peroxide. Generally, various parameters were altered by application of hydrogen peroxide like relative water content, photosynthetic rate, stomatal conductance and transpiration rate of soybean plants. So you see that how hydrogen peroxide is modulating very significant metabolic parameters of the plant. Now, what is the role of hydrogen peroxide in salinity stress? So you see that hydrogen peroxide also plays a very critical and vital role in enhancing plant resilience to salinity stress by serving as a sort of signaling molecule. You see that it regulates ion homeostasis, helping to maintain a balance between sodium and potassium ions, which is very critical for cellular functions under salt stress. Hydrogen, in addition to this, hydrogen peroxide also mitigate oxidative damage by scavenging excessive production of reactive oxygen species through activation of antioxidant defense mechanism, including enzymes like superoxide dismutase, catalase. And in addition to this, you see that in one way it is inducing the antioxidant enzyme, it also promotes the synthesis of osmoprotectant. This is also very crucial, such as proline and glycine betaine, which generally stabilizes cellular structures and maintain a sort of osmotic balance. Additionally, hydrogen peroxide also interacts with hormonal and signaling pathways, including abscisic acid and nitric oxide, to activate salt tolerance gene and stress responsive pathways. So these multifaceted roles underscores the importance of hydrogen peroxide in plants against the detrimental effects of salinity stress while supporting growth and metabolic functions. Now this slide shows a simple graphic depiction of the main molecular mechanism in response to plants treated with hydrogen peroxide and how they could be improved salinity stress tolerance in plants. So this table basically provides information about the concentration and timing of hydrogen peroxide priming in plants along with its positive impacts and mechanism in reducing the salinity stress. It highlights how optimal level of hydrogen peroxide at the right time improve the ion balance, enhance antioxidant defense and promote osmoprotectant production and activate stress responsive pathways, all of which is helped plants to adapt to and withstand the harmful effect of salinity stress. So from these findings, you just have a look that how hydrogen peroxide is mitigating various stresses, particularly salinity stress. Now, what is the role of hydrogen peroxide in temperature stress? Why I am elaborating all these role of hydrogen peroxide in all these stresses? Because this is a very versatile molecule, and you see that its use could be implemented in any sort of abiotic stresses. That's why just I am giving a brief overview of all these stresses in nutshell. So in addition, in continuation, what is the role of hydrogen peroxide in temperature stress? You see that hydrogen peroxide basically modulate heat shock proteins during high temperature, and hydrogen peroxide also is very crucial for protecting photosynthetic machinery from damage to heat stress, and it also regulates cold responsive genes under low temperature, and in general it enhances antioxidant enzyme activities and thereby it aids in maintaining cellular stability under extreme temperature. So this is the role of hydrogen peroxide in temperature stress, just I discussed in nutshell. Now this is the model that how H2O signaling cascade is triggered under low temperature stress. This is a very relevant figure. If you go through the figure, you see that under low temperature stress, what happened? Hydrogen peroxide acts as a signaling molecule and initiating various defense responses in plants. It triggers an increase in the intracellular calcium levels, which acts as a secondary messenger to activate downstream pathways. The mitogen activated protein kinase cascade basically is then activated, amplifying the hydrogen peroxide signals, where you see that H2O2 hydrogen peroxide also interacts with glutathione pool and converting reduced glutathione to its oxidized form, then which modulates redox homeostasis. So this redox signaling in turn activates transcription factors, and these various genes encode proteins which enhance cold tolerance by stabilizing cellular structures and maintaining metabolic functions. So in addition to this, hydrogen peroxide under cold stress condition modulates activity of catalase, balancing the production of ROS level to prevent oxidative damage while ensuring effective signaling at the same time. So through these intricate networks, hydrogen peroxide orchestrates, or you can say involved in plant responses to low temperature stress, enhancing the survival of the plants and its further adapted mechanism. Now, this is the figure just my student published one of the review paper on hydrogen peroxide. So, in nutshell, what is the role of hydrogen peroxide in heavy metal stress? We can have a quick look that the generation of reactive oxygen species, including hydrogen peroxide, activates a complex signaling network that helps plants to respond and adapt under heavy metal stress. Basically, as I mentioned that hydrogen peroxide acts as a signaling molecule by interacting with specific receptors and redox sensors such as calcium channels and cysteine kinases, which initiate a cascade of intracellular events. And this includes the activation of the mitogen activated protein kinase pathway. I'm amplifying the stress signals. So basically hydrogen peroxide modulates the redox status of the cell, interacting with antioxidants like ascorbate and glutathione, regulating oxidative stress and preventing cellular damage. So these interactions, along with the formation of various regulatory proteins, lead to the activation of transcription factors which promote the expression of genes responsible for metal detoxification, antioxidant defense and cellular repairs. So this is a model showing how hydrogen peroxide could protect plants under heavy metal stress. In addition to its direct role, hydrogen peroxide interacts with other plant growth regulators such as abscisic acid, auxins, cytokinins. For example, you see that hydrogen peroxide enhances the signaling of abscisic acid, which helps to manage loss of water and promotes stress tolerance under drought stress. Then hydrogen peroxide also crosstalk with auxins, cytokinins to modulate growth and development under abiotic stresses. So basically these interactions along with the hormone, other hormone, fine tune the stress response of the plant, balancing growth and defense mechanism. Therefore, hydrogen peroxide works along with other plant hormone and signaling molecules to just modulate the multifaceted response of plants to heavy metal stress, improving tolerance and survival of the plant. So this is a very crucial figure. Now this is the table showing the ameliorative role of hydrogen peroxide in heavy metal stress. So if you want to have a quick look that how hydrogen peroxide could be applied and at what concentration it should be applied to the plants to generate positive response. So from this table you can have a quick look. Now just see that how hydrogen peroxide interacts with multiple phytohormones, creating a complex network of signaling pathway that regulate key plant functions. This crosstalk influences processes like formation of root, germination of seed, stress tolerance and cellular response to abiotic stresses. So if I talk about what is the role of hydrogen peroxide in formation of root, basically hydrogen peroxide interacts with auxin to regulate initiation of root, whereas auxin promoting production of hydrogen peroxide, which in turn facilitates the formation of root. So how hydrogen peroxide interact with gibberellins? So hydrogen peroxide basically modulates biosynthesis of gibberellic acid by influencing enzymes involved in production of gibberellic acid, promoting seedling growth and under favorable and under unfavorable conditions. So hydrogen peroxide also reacts with cytokinin, it also reacts with brassinosteroid. And Andrzej has already discussed detail the role of brassinosteroid under abiotic stress, so I am not going to repeat it. And hydrogen peroxide also interacts with jasmonic acid and activates antioxidant system. So you see that hydrogen peroxide at low concentration does not act in solo. It also interacts with other plant hormones. The motive to show this slide is that hydrogen peroxide does not act in solo. It interacts with other plant hormones to carry out number of specific plant growth and developmental functions. Now, moving ahead, you see that hydrogen peroxide also interacts with other hormones like nitric oxide. Yesterday, Dr. Gauzia Bashiri discussed a detail regarding the role of nitric oxide. So, my point is that hydrogen peroxide also interact with nitric oxide and calcium ion, and which are very crucial signaling molecule in plants that regulate physiological processes, development and various stress responses. You see that various abiotic stresses trigger the production of hydrogen peroxide, nitric oxide and calcium, and hormonal signals like abscisic acid can also act as a stimuli. And basically nitric oxide enhances activity of NADPH oxidase, leading to increased production of hydrogen peroxide, and then together they regulate calcium signaling, stomatal movement and transcription factor activation. Hydrogen peroxide also modulates calcium influx by opening calcium channels present on the plasma membrane. This leads to a spike in the cytosolic level of calcium, which acts as a secondary messenger. So, in addition to this, mitogen activated protein kinases are very central to integrating signals from hydrogen peroxide, nitric oxide and calcium. These mitogen activated protein kinases basically transduce signals to regulate gene expression, activation of enzyme and production of defense molecule. So, these cascades basically influence the plant growth like root elongation, leaf expansion and various stress responses like production of osmolytes and activation of antioxidant defense system. So, this is the figure showing the retracted from one of my published paper in Chemosphere this year in 2025, and this figure talk about a crosstalk of hydrogen peroxide with polyamines and nitric oxide and calcium during abiotic stress, and how mitogen activated protein cascade is responsible for activating various responses in plants. So, various abiotic stresses generally could trigger the level of polyamines, just I discussed in detail in my previous lecture. An elevated level of polyamines and calcium basically promote elongation of root, enhancing tolerance to abiotic stresses such as drought and salinity. So, this rise in the level of polyamines basically stimulates one of the important crucial enzyme, polyamine oxidase, leading to generation of hydrogen peroxide. So, here hydrogen peroxide come into play by increasing the level of polyamines. So, this sort of interaction with polyamines, hydrogen peroxide and calcium activates various reactive oxygen species signaling pathways and increases activity of mitogen activated protein kinase. So, this in turn basically induces the production of transcription factors that regulate the expression of stress tolerance genes, thereby enhancing the resilience of plants to various abiotic stresses. Now, I will just in a quick, I will show you some of my case study dealing with the interactive role of epibrazolide and hydrogen peroxide in regulating stomatal physiology, root morphology, photosynthesis and growth rates in Solanum lycopersicum under nickel stress. So this slide shows the effect of various treatment of hydrogen peroxide, one of the brassinosteroid in combination to nickel stress plants, and how these hydrogen peroxide, EP brassinosteroid in solo or in combination affected the growth of the plant grown under nickel stress. So you see that how the plant morphology, you can visualize it change with those plants which were growing under nickel stress, and then application of low concentration of hydrogen peroxide and ipobrassinosteroid mitigated the damaging impact of nickel. Now so you see that if plants are grown in any sort of heavy metal stress, it is going to damage the morphology of the root. So in this case also the morphology of root was severely affected by the plants supplied with nickel. So once the plants were given treatment of low concentration of hydrogen peroxide, it generally restored the morphology of the root, and it is very much clear from this slide. Now this figure shows how the photosynthetic parameters were altered by nickel, and you see that low concentration of hydrogen peroxide was very effective to overcome the nickel stress. You see in this slide that how brassinosteroid and hydrogen peroxide help to mitigate the nickel-induced changes in antioxidant parameters. Now this is another case study from my lab where role of hydrogen peroxide in modulating photosynthesis and antioxidant defense system in tomato berry studies where plants were given copper stress. In my previous studies it was nickel stress, here it is copper stress. Now you see that how stomatal responses were altered by plants given with nickel stress. The high concentration of copper led to closure of stomata, but once low concentration of hydrogen peroxide were given to the tomato plants, it restored the normal morphology of stomata. Now one of the interesting methodology I would like to discuss what is the role of diaminobenzidine and nitrobutetazoline, and these two chemicals basically they are widely used as a tool to detect reactive oxygen species in stress plants. So these two dyes, basically diaminobenzidine and nitrobutetazoline, they are used as a tool to detect reactive oxygen species in stress plants. So their basically mechanism is based on their ability to visualize oxidative stress by staining a specific reactive oxygen species such as hydrogen peroxide and superoxide radicals. So basically this diaminobenzidine specifically detects accumulation of hydrogen peroxide. So in this context this diab dye is very significant. It can detect and bind the hydrogen peroxide. It basically reacts with hydrogen peroxide in the presence of plant peroxidases to form a reddish brown polymer which appears as a distinct stain on the plant tissues, and these reactions highlight the regions of H2O2 production often corresponding to areas under abiotic stress such as drought, salinity and heavy metal stress. So likewise if I talk about another dye, nitrobutetazoline, basically this dye detects superoxide dismutase radical by reacting with the superoxide to form formazole, and this formazole as insoluble blue compound. This blue color marks region of the superoxide accumulation which often occurs in organelles like mitochondria, chloroplast, plasma membrane under stress condition. So these staining methods are basically very much instrumental in students carrying out research on abiotic stresses, as their visualization and localization of ROS in plant tissues is very crucial. Just I mentioned that DAB staining is very crucial for visualizing the accumulation of hydrogen peroxide, whereas NBT is very significant for visualizing the superoxide radicals. So just to have a look of how these two dyes, they are significant and playing a role to detect superoxide radical as well as hydrogen peroxide. That's why I discussed it here. Now you see that how these accumulation of hydrogen peroxide and superoxide radicals was detected. This slide clearly shows it. Now I would like to conclude my lecture with that hydrogen peroxide exhibits a dual nature in plant cell. At high concentration, it causes oxidative damage and like lipids, protein and DNA, potentially leading to cell death. However, at low concentration, hydrogen peroxide functions as an important signaling molecule involved in regulating plant responses to various abiotic stresses. In addition to this, hydrogen peroxide also plays a significant role in plant responses to abiotic stresses and it participates in systematic signaling and may regulate various physiological processes, helping plants to adapt to various environmental challenges. Hydrogen peroxide signaling is basically closely integrated with other signaling networks in plants, allowing for the coordinated regulation of developmental processes and stress responses. So this integration ensures that plants can efficiently manage their internal responses to changing environmental conditions. Now, what are the future prospects? The exact site of hydrogen peroxide production under different stress conditions are still a matter of debate. The identification of dedicated sensors or signal transducers complex for hydrogen peroxide related proteins is still lacking in plants. The direct role of hydrogen peroxide as an activator in stress responses remains still not clear. Understanding the correlation between hydrogen peroxide oscillations and other signaling pathways will be a major research focus. Additionally, determining the specificity of hydrogen peroxide and how it interacts with other stress-related pathways will remain a key challenge for plant biologists in coming years. Thank you for very much for listening my lecture. If you have any query, just you are free to ask and raise. Okay. Okay, thank you very much.
today's tutorial, we are going to discuss that how we can use IRGA. So, full form of this machine is infrared gas analyzer, and it is one of the most, or we can say, very important instrument in plant physiology, as by using this particular instrument, we can measure several parameters which are related to photosynthesis and gas exchange. So basically, IRGA can measure net photosynthetic rate, internal CO2 concentration, stomatal conductance, transpiration rate, as well as water use efficiency. So just to, in short, we'll discuss that how it works and what is IRGA, what are its components. So we are having Liquor 6400. Basically, it is a sophisticated portable gas exchange machine which is designed to measure photosynthesis and gas fluxes in plants. It has been widely used in plant physiology research due to its high accuracy, flexibility, and ability to stimulate various environmental conditions. Gas exchange techniques can be used by using IRGA as it employed for decades as it measures carbon dioxide and water fluxes in leaves, and occasionally we can also use it in non-foliar tissues. Apart from these, it can also measure leaf physiological performances and photosynthetic capacities, offering insights into carbon and water fluxes in ecosystem functions. So, we should have a basic idea that what is maximum capacity, what is flux, and what is conductance. So, basically, maximum capacity is the intrinsic photosynthetic potential of a leaf. It indicates maximum ability to fix carbon dioxide, as which it can be used to conduct photosynthesis in ideal condition. What is flux? Flux denotes rate at which carbon dioxide or water vapor moves in plant tissues during photosynthesis and transpiration respectively. It provides insight into dynamic exchange of gases between the leaf and atmosphere. This is the major principle that it basically helps in measuring the exchange of gases between leaf and atmosphere. Net photosynthetic carbon dioxide assimilation, stomatal transpiration, and respiration rates are common fluxes which can be measured. What is conductance? Basically, conductance is described as a rate at which gas passes through stomata or leaf mesophyll tissues. We all know that basically the function of stomata, it helps in gaseous exchange. So by using this machine, we can measure that what is the stomatal conductance or what is the internal CO2 concentration in our sample. It can also reflect opening of stomata affecting both carbon dioxide uptake and water loss through transpiration. Basically, conductance is a key regulator of photosynthetic efficiency and water use strategies in plants. So, what is the procedure or processing of IRGA? Basically, it has various components. So, firstly, it has infrared absorption principle. So, what is infrared absorption principle? It is carbon dioxide and water vapor molecules which absorbs infrared light in a unique specific wavelength. Secondly, the amount of absorbed infrared radiation is proportional to concentration of these samples, as it's obvious that infrared radiations and gases which are, or the concentration of gases are inversely proportional. For say, if infrared radiation is high, so gases will be high. And secondly is the dual path detection system. This Liquor 6400 employs two separate gas pathway. First one is sample path and second one is reference path. Obviously, we need reference as it has ambient air for comparison and it has unaffected leaf gas exchange parameter. And in sample path, basically it is our sample which we are going to use, and it is used basically to measure air from the leaf chamber where the plant is exchanging gas. This dual part design helps to correct for environmental fluctuation and it improves measurement accuracy. As we know that we are having a lot of environmental fluctuations, so this reference path helps to deal with this problem. Moving ahead, next one is gas exchange monitoring. So basically here, air containing carbon dioxide and water vapor from that leaf chamber flows through IRGA gas analyzer. The analyzer then detects the difference in infrared absorption between the sample and reference air. As you can see that in this diagram, it is clearly shown that basically it measures difference between reference sample and in sample IRGA. Next one is changes in CO2 concentration provide information about photosynthetic rates, while changes in water vapor concentration reveals transpiration rate. So moving ahead, next one is photosynthetic rate calculation. Basically this system helps to measure net photosynthesis. It can be measured by using reduction in CO2 concentration between the incoming and outgoing air from the leaf chamber. As you can see that air in and air out, this can be measured and we can easily measure net photosynthetic rate, internal CO2 concentration, instrumental conductance and transpiration rate. Next is water vapor and transpiration movement. Water vapor concentration are measured alongside carbon dioxide, enabling the estimation of transpiration rates. These measurements are crucial for evaluating water use efficiency under various environmental conditions. As we know that when plants are subjected to any kind of stress, then majorly photosynthesis and gas exchange parameters are hampered. So in previous tutorial, we have discussed about junior PAM. Junior PAM is basically used to measure for PS2 efficiency or PS2 activity. We can measure FV by FM value from there. But here we can measure net photosynthetic rate, internal carbon dioxide concentration, stomatal conductance, water use efficiency and transpiration rate. So this is most important instrument and luckily our lab is having it and it has helped a lot in our research. So what is the working principle of this IRGA? The gas exchange measurement using this LICOS 6400 IRGA operates as an open system. It is basically an open system. The system used two infrared gas analyzer to measure the concentration of carbon dioxide and water. One before the air enters the chamber as its reference IRGA and another one is exit as sample IRGAs. By comparing these values and knowing the air flow and enclosed leaf area, the system calculates critical plant physiological parameters such as carbon dioxide assimilation, transpiration. This data helps to assess photosynthetic efficiency and water used under several environmental conditions. Basically, this figure is showing that this is the principle of IRGA, as I have told you earlier also, that IRGA operates as an open system. The concentration of carbon dioxide and water vapor which is incoming and which is also termed as reference IRGA alongside with the air flow entering the leaf chamber, these parameters are then measured again by sample IRGA. So we will discuss it while I will demonstrate it that how it functions. So what are its application? So as you know what are its application, but in brief I am discussing it with you. Basically it evaluates photosynthetic efficiency under different environmental conditions. It studies the impact of light, temperature and carbon dioxide level on photosynthesis. Apart from it, it also analyzes plant responses to stress factors such as salinity, drought, heat and nutrient deficiencies. It also helps in selecting plant varieties with improved photosynthetic efficiency and stress tolerance. At last, it also assesses carbon assimilation and water use pattern in natural ecosystem. So what are its advantages? It have many advantages. So just in short, I will discuss. It is highly accurate and sensitive to carbon dioxide and water vapor measurements. Secondly, it operates as dual part design. As I have told you, it has reference and sample IRGA. Third one is it is portable and suitable for field studies. And lastly, it have real-time data acquisition and advanced analysis capacities. So basically, this is completely assembled IRGA, which is used in field to measure various parameters. So I'll now demonstrate you that how this IRGA is used. So first of all, these are the So we are having different components of IRGA. So this is IRGA unit. You can see here, it is having display. So everything which we are going to select or deselect, everything is done on this main unit. This one is leaf chamber and it have a sensor here, and after when it's open, it also have thermocouple sensor here where we will be placing our leaf. Secondly, this one is tubing system. Basically, this tube is going to assemble in this inlet and outlet port, which is going to connect this leaf chamber and with this main unit of IRGA. Now, as you can see that these both units are de-assembled. Now, firstly, we'll assemble it. Assembly of IRGA is quite tough and it should be done meticulously because to make its airflow regular. So first of all, we are going to assemble this leaf chamber. This one is going to assemble here in this port particularly. Secondly, we are going to assemble these ports in these inlet and outlet ports. Basically, this helps in regular airflow. Now, this should be assembled in this port. As you can see, here are wire and here are holes. Now, very meticulously, we have to tighten these screws. Ensure that tightening should be done so that airflow is measured here or there is no leakage of air from here. Now, this particular leaf chamber is assembled with this tubing. Now, this leaf chamber is going to connect with this main unit. As in this main unit, you can have a look that we are having these ports, these inlet and outlet ports. So obviously, these two will be connected in these two ports, and these two will be going to be connected in these two. As you can see that this black one going to be connected with this black. Here is the sign of this black, and this one to this port. Assembly of this IRGA is quite tough. Apart from this assembly, basically this helps in airflow. as we are not doing it in field conditions, so there would be hindrance in reading. Just you have to learn how to assemble it and how to measure readings from it. This is battery, so now I am going to connect battery with this port. So now our IRGA completely connected. If we are using it in field conditions, we can use this stand. Is this IRGA is having here where we can adjust this stand, but now we are using here in classroom, so there is no need to adjust this stand. So here we are having this switch. We'll switch on it. It will take some time to warm up or to stabilize. So we have to wait for 30 to 40 seconds. Again, this is controlled, this one is stressed, and this one is hormone treated. Fine. So now you have that idea. In stressed condition, photosynthetic or gas exchange parameters will be low. However, in hormone treated, these will be high. So let's see that what we are getting in it. So you cannot see in this display we are having three options. One is Arabidopsis chamber, second one is default fluorometer, and third one is factory default. Just you have to enter here because we want to use factory default system. Enter. Now processing is going on. Now they are asking, is the chamber IRGA connected? So you can see that chamber is connected. So there is Y or N. Y stand for yes, N for no. Just you have to click, select here Y. Again processing is going on. Now options which are coming here are welcome menu, configuration, calibration menu, new measurement, and utility menu. Obviously we have to take new measurement. So for new measurement is at F4 option. For example, welcome menu at F1, configuration at F2, calibration at F3, and new measurement at F4. So we have to select F4 button here. Now it is stabilizing itself. Fine. So we have to wait for few seconds here also. Now readings are in positive. We have to wait that for it to stabilize and become positive. So what are all readings are coming here are photosynthesis, CI is internal CO2 concentration, TR millimole, that stands for transpiration, then is relative humidity and all those parameters, you don't need it. So yeah. Now reading is it in positive. Basically you can see any one of the reading. As I am seeing photosynthetic reading, now it became positive. So we are ready to use it. Now how we can operate, or we have to click on add remark. Add remark is at fourth option, F4. Or we can say that we can click on F1, open log file. Then after open log file, we can name this file. I am giving this file name as Gian, date 13/2/25. Then again you have to enter here. Now here is enter or edit remarks. Now what do you mean by enter or edit remarks? It means your treatment pattern. For example, if this control 1 is treatment 1, this stress 1 is treatment 2, and this hormone 1 is treatment 3. So T1, T2 and T3. Now you should note this in your files that which one is T1 or which one is T2 and so on. So I am naming it as T1. So just you have to open very meticulously this IRGA. You have to insert you have to remove this sensor as it detects sunlight, but here we are not having sunlight, so just I am showing you. Now you have to close this. Your leaf is inserted here in this leaf chamber. What you have to do, as I have already done T1 here, You just have to click on enter and wait for your reading to get stabilized. As readings are fluctuating, obviously we will not get readings what we can get in field condition because there is no sunlight, and on sensors we are having no sunlight here. For example, now we are getting photosynthetic reading as 13, so we can take this as reading. So you have to click 3 times on F1, then F4, again you can get next reading as it get detached because now again by entering T2 you can calculate next parameter. This one is T2. Now enter here, just wait for reading. The reading is in minus. Obviously photosynthetic will be never it will give its reading in minus, so we have to wait till it gets stabilized. Now it's giving its reading as 9, so we have to wait 3 times, press F1, then F4, then again same option came. Now we have to enter T3. We have to close it, we have to, now this one is T3, hormone treated. Just make sure that whole leaf sample is placed under this leaf chamber because inside it are having thermocouple sensors, okay? And there should be no dust particles or water in it, neither our reading will not come. And it can also damage our sensor. Again, now I am clicking or selecting enter. We are getting 24 here. So okay, fine. 3 times F1, then F4. So these are the readings. So now you may come in group of 10. Come this way. Plant to liao. Stuff here. What's here? T3. Just you have to mark T4 here. Then open this chamber. ये डिटैच हो गया है, इसको यहाँ पे इंसर्ट करो हल्के से, करना है, करो इसको बंद करो, यहाँ पे एंटर कर दो, रीडिंग्स आ जाएंगे. Here we are having photosynthesis. You can see here clearly CI, transpiration. These are other parameters, flow meter. As you can see here, we are having S, CO2 S. S is for sample and R is for reference, as I have shown you earlier. Fine. So now reading is in negative. Just click on F1, F4. यूज करना है. बंद करो. बंद करो. T4 माइनस है 19.9, वो 19.9, ठीक है. T5 next. Clear? You have to close it. After operating it, you have to close it. Neither your readings will get interfered. because F1, F2, F3, F4. Basically, F1, F2, F3, F4 and F5 keys coincide with these blocks, or we can say measurements. For example, on F1 side we are having log del in, on F2 we are having view clear, and on F3 we are having close delete chance, and on F4 we are having add caps lock. So if we want to select add caps lock, so we have to select key F4. There is no such tough task because this is not touch. If this is touch, then we can only touch and measure our. This one is photosynthesis. This one is internal CO2 concentration. This one is transpiration rate. Fine. This one is CO2 of reference. This one is having CO2 of sample. H2O water of reference. H2O water of sample. Fine. Clear? You can treat it with any kind of stress, any abiotic or biotic stress. For you can take any kind of stress. For example, you know that we are having biotic and abiotic stresses. As our work is on abiotic stress, so you can take salt stress, heavy metal stress, drought stress, whatever you want to take. High temperature stress, low temperature stress. It's up to you, whatever stress you are taking. This plant is treated with chromium. Fine, heavy metal stress. And it completely depends on concentration, that how much concentration you are applying. More the concentration, more negative effect you will see. Fine. Okay, fine. These are basically bypass scrub. This is for basically this one is tightened, this one is for reference, and this one is for sample. You should not touch it. You should never touch it because it is already calibrated. If you move it or roll it, then readings will get hampered or interfered. As it is calibrated, so you should not touch it. You just have to operate here. You should measure readings here only. So EGA is done. You should wait for some time to stabilize it. You can take it F, press 3 times F1. How? बंद करो, प्रेस करके बंद करो, लॉक करो. आई ना आवाज. दिस इज फॉर लॉक. ठीक है ना, पकड़ो. ये है ना रीडिंग. ऑब्वियसली अब यहाँ पे हैम्पर हो रही है क्योंकि लाइट नहीं पड़ रही है, पीछे जा रही है. कुछ नहीं यहाँ पे. 3 बार F1, then F4. नेक्स्ट सैंपल के लिए ये सारे पोर्ट्स हैं यहाँ पे, ये ऑन एंड ऑफ बटन है। बंद करो, बंद करो। बंद कर देना है इसको। हाँ करो, बंद करो। लॉक हो गया, पकड़ो, पकड़ लिया। अब इसके बाद यहाँ पे ट्रीटमेंट 6 एंटर, रीडिंग आ रही है। अब एक बार जब ये स्टेबलाइज हो जाए, you have to wait for few seconds to stabilize. F1, then F4. Again, your readings will be noted. ठीक है? इसको बंद करो, बंद हो गया। Another most important thing is that you have to clean it regularly. As you can see that it is having dust particles on it, so you have to regularly clean it so that neither your readings will get hampered or interfered. एक बार इसमें कर, एक बार खोलो, लीव रखो। रखने के बाद एंटर। ठीक है, यहाँ देखो रीडिंग है अभी माइनस में, अभी ये पीछे जाएगी, स्टेबलाइज होगी, जा रही है माइनस में। ये आ गई हमारी 22.9। ठीक है, निकालो, बंद करो। ठीक है, आगे समझ तुम उसमें लो। खोलो, खोलो, लगाओ, बंद करो। अब यहाँ पे T7 लिख के एंटर करो। ये वाली जो रीडिंग है ये फोटोसिंथेसिस की है, कंडक्टेंस की है, इंटरनल CO2 और ट्रांसपायरेशन की है। ठीक है, अब इसके बाद क्योंकि अभी बहुत ज़्यादा इसलिए आ रही है क्योंकि लाइट नहीं है प्रॉपर। जब इस पर लाइट होगी तो ये स्टेबलाइज हो जाएगी। अब जैसे ये आ गई यहाँ पे, इसमें 3 बार F1 कर लो, F4 कर लो। F1 3 बार इसीलिए कर रहे हैं कि वो रेप्लिकेट दिखा देगा हमको। ठीक है, बैक कर दिया, F4 क्योंकि हमें बैक जाना है, फिर दोबारा दोबारा थोड़ी खोलोगी, बंद करोगी। ठीक है, खोलो, इसको बंद कर, एक बार खोलो, खोलो, बंद करो। क्या है यहाँ पे, पहले देखो, ट्रीटमेंट 7 है ना, बैक करो, ट्रीटमेंट 8। ठीक है, करो। बाप रे, ठीक है, फिर एंटर। देखो सब आ रहा है ना, फोटोसिंथेसिस, कंडक्टेंस, इंटरनल CO2, ट्रांसपायरेशन। ऊपर जो है these are for reference sample, relative humidity, CO2, H2O2 in millimole, फ्लो मीटर आ गया। अब जैसे मान लो 21 माइनस में नहीं है बेटा, ये देखना है ना फोटोसिंथेसिस। अब देखो मैंने एक बार F1 कर दिया, तुमने बोल दिया F4 नहीं हुआ, रीडिंग खराब। देखो कितनी ज़्यादा चली गई। ठीक है ना, तो इस चीज़ का इसमें ध्यान रखना है, इसीलिए
हम सारे मेजरमेंट्स ले रहे हैं, तो ऊपर की साइड जो होता है प्लांट में, वो ट्रांसलेशन होता है।
ऊपर नहीं, सेकंड मोस्टली सेम है। ये यहाँ पे देखो ना, ये प्लांट में हम नीचे नहीं ले रहे हैं, ये ऊपर ही ले रहे हैं, ये बड़ा प्लांट नहीं ले रहे हैं।
तो मतलब कोई सा भी रैंडम
नहीं नहीं, नीचे वाली नहीं लेनी, ऊपर से सेकंड नोड वाली लेनी है।
आइडियल
एग्जैक्टली, जैसे इसमें इसकी लो, आ गई ना चीज़ समझ में। कुछ नहीं, सेव करना है F1, F4, फिर देखो बैक में आ गए, यानी अब नेक्स्ट पैरामीटर के लिए सकते हैं, ठीक है। F4 और F1 का कॉन्सेप्ट कुछ नहीं है यहाँ पे, यानी अगर लॉग उस पे जाना है F1, व्यू पे जाना है F2, इस पे जाना है, मतलब उसके नीचे वो कंसाइड कर रहे हैं, क्योंकि ये टच नहीं है ना, टच होता तो ऐसे कर देते है। चलो भाई बंद कर दो, बस बस बस। सुबह में you have to record readings of IRGA on bright sunlight. From 9 to 11 is the most ideal time. AM, obviously. ठीक है, 12 आ गया, यहाँ पे जो है तुम्हारी CO2 कंडक्टेंस आ गई, ये सब रीडिंग्स आ गई, ठीक है। बंद करो, लाइट ही नहीं है ना यहाँ पे, कुछ भी हो सकता है। आओ नेक्स्ट, so now I think you all have idea that how to operate this. Now just we are exiting from it. जी जी, thank you so much.
Thank you, Dr. Yamshi, for excellently demonstrating the working of IRGA unit. And I hopefully all of you have learned it, how to use the IRGA. So next, we will have Dr. Professor Masroor Khan for the demonstration of how keratin is synthesized in apparatus. So he will demonstrate in a while. So you have to wait for the some time.
laboratory demonstration. PPT. Slide is move. बस वाला है लिख वाला करा दीजिए जे में डाल दे Well, Assalamu alaikum and good afternoon to all of you. Yesterday, I gave a talk on smoke water and kerikins, how these smoke water and kerikins can be used to enhance the seed germination of plants, as well as the smoke water and kerikins can be used to increase the growth of plants and yield and quality of the plant. So today I would like to give a demonstration about the equipment which is used for generating smoke water. So this equipment has been prepared by myself, and I was induced by the history of kerikins, as I told you yesterday, that in Australia, in jungles, the fire forest was a common phenomena, as in recently in California, the jungles were burned and huge loss of property and forest was done. So the scientists examined in Australia that next year of the fire forest, there was a very luxurious germination of plant and very healthy growth of the plant was found. So they found that smoke, particularly the smoke formed by the burning of twigs, leaves and wood, that created some chemicals which are known as kerikins. So these kerikins are helpful in increasing the germination of plant as well as the general performance of the plant. Okay. So I read the whole story and then I prepared this equipment by myself. So first I purchased a suction pump from USA. That was not very expensive. And then I purchased a hot plate from Mumbai, and then the rest of the setup was prepared over here. So unfortunately, the suction pump which I purchased from USA was defected and it was not working. So I purchased a new suction pump today, and instead of using a hot plate, today I am using induction plate or induction cooker. You are familiar with this induction cooker because this works only on the steel utensils. Okay, so only on the steel it works. Otherwise, aluminum or any other material it will not work. Okay, ceramic or aluminum or other copper like that, it will not work. So for this purpose, this is more cleaner than using the hot plate. I purchased a induction cooker, induction heater, and then I took a utensil of steel as a bowl to hold the straw, because I am preparing the smoke from the straw, wheat straw. Wheat straw were purchased from the market, and then I washed it, dried it in the oven, and all the moisture was removed in the micro oven. So the whole straw was completely dry. When it was dry, I poured into the steel utensil and covered with a funnel, and the funnel was connected to the suction pump. Okay. The funnel was connected to a flask, a suction flask, okay, in which the glass tube was gone to the bottom, and the flask was filled with the DDW, okay, and the pump was turned on. suction pump was turned on, and then the smoke generated by the straw went through the flask. Just like a technique of hookah. Are you familiar with hookah? Have you seen it? So the almost same principle is applied over here, and this is very old tradition of hookah. I have also seen in Egypt, the people are very much fond of hookah, especially in the meetings. They like it very much. So similarly, the smoke was sucked by the pump, and the smoke was carried to the suction flask, and the smoke was dissolved in water. Okay. So first we turn on the induction heater, and the charring of wheat straw starts, and when the charring starts, it generates smoke. The smoke was sucked by the pump through the water. Okay. So the water will become yellow in color. Gradually it will be yellow in color. So after 5 minutes of operation of the equipment, we will take a sample by using a micropipette, and we take only 0.5 ml, that is 500 microliters of sample, and we will read in the spectrophotometer at 430 nanometers. Okay, 430 is the best optical density for reading the smoke water. Okay, so obviously the optical density will increase time to time. So after each 5 minutes, we take a sample of smoke water, we read the optical density, and a time will come when no more smoke will be dissolved in water. So this is called as SSW. When optical density is not increasing, it means that double distilled water is fully saturated with the smoke. Okay. Now you can use this SSW, that is smoke saturated water, for your experiment. Obviously, you have to dilute it because it is highly concentrated, and kerikins are required in very minute quantity, -8, -9 molar concentration. Very minute concentration is effective in inducing the seed germination and inducing the plant growth. So you have to dilute it. In our experiment, we diluted it 1 to 1000 dilution, 1 to 500 dilution, 1 to 250 dilution, and 1 to 100 dilution. Okay. So obviously 1 to 1000 dilution was the most dilute, and 1 to 100 dilution was the most strong, or the most concentrated, you can say, right? So we sprayed this diluted, saturated smoke water on plants, and different parameters were studied, and we found out that in most of the cases 10 power minus 8 m was the best concentration of smoke water in increasing the yield and growth and other performance, and also seed germination. Okay. In some cases 10 power minus 9 m was also proved best. Parallel to this, we also worked on Carikin. Carikin was purchased from Alberta Chemical Agency from Canada, and this is very expensive chemical. As I told you that 1 milligram it costs 25,000 rupees. So for the sake of comparison, standard is must. So we purchased this chemical from Canada, 1 milligram Carikin KR1, and then we compared it. Also, we studied the effect of KR1 on the plant, and we took 4 concentration, that is 0, 10 power minus 9 m, minus 8 m, minus 7 m, minus 6 m. Okay. So in this way, we also studied the effect of smoke, water, and as well as effect of Carikins, right? So today I am going to demonstrate how it works, okay, and how you can prepare this apparatus, okay, at your own cost. So these are different parts of this smoke generating plant. This is induction heater of Philips company. So first you have to start at the full speed, then you can reduce it up to minimum, okay? And then smoke will be generated by the heating of this straw. Inside this utensil, there is straw. We first took the straw from a village, and then we washed it and dried it in oven, and when the water was completely removed, then we transferred the straw in this steel utensil, right? And then it was placed on the top of the induction heater, right? This was covered by a funnel, receiving funnel, okay? This funnel was connected by a plastic tubing. Tubing was carried to the suction flask, the suction flask. So this tube was connected to a glass tube, okay? Okay, and in the glass tube, the lower end was just above the bottom. So it should not be touched with the bottom, but rather 0.5 centimeter or 1 centimeter maybe above, so that the smoke can pass easily, right? Then we take the double distilled water in the flask, suction flask, and it should be measured. It was measured नहीं किया हमने, but you should take 1 liter of, exact 1 liter of double distilled water inside this suction flask, right? Now this is suction pump, okay? This is made in India, and this suction pump, basically this has a dual function. If you connect the connector with this point, this will suck the air, and if you connect the connection with this point, this will push the air, okay? So dual function. So now I am using this equipment for sucking the air as a vacuum pump, right? And good thing is that this also contains a regulator, okay? You can control the speed also. For example, 1, 2, 3, 4, 5, 6, up to 6 level, you can increase it. But normally we run it at either 2 or 3 point, okay, of suction pressure, right? So this is a power supply, and so now we start the induction heater. I think this is the connection of heater, and then we have to turn it on. This is power button, and then we have to deal it with a manual system. So first make it very fast heating like this, deep fry, that is about 200 degrees centigrade. Now you can turn it off, and then you can decrease the temperature to the minimum level. Turn it on, go to manual, and then you can decrease temperature to the minimum. It is 120 degrees centigrade temperature, right? So now it is burning, and the smokes are slowly coming out, and it will advance also. And then we'll turn on this vacuum pump, and here is the button for the power supply, and then we have to make it turn it on. You can lock it also. You see? You see, smoke is forming, and now it is going to the water. So now the smoke is being dissolved in double distilled water. So it will become yellow. After each 5 minutes, you have to turn off the system. After 5 minutes, you have to turn off the system like this, and then after 5 minutes, turn on the system, and then take a micropipette, fix the reading at 500 microliters, and then open this bottle. This is attached with a cork. Open this like this, and take a sample from this water, and like this. Okay. So anyway, just we have to take 500 microliter of the sample, transfer into a spectroscopic tube, and read the optical density at 430 nanometer. Right? Because 40 is the optimum maxima. This is the reading where the maximum OD comes. So we have to read all the readings at 430 nanometers. In this way, you will find a graph. After 5 minutes, 10 minutes, the optical density will increase. A time will come when there will be no increase of dissolving of smoke in water, and this will be fully yellow in color. Okay? No change in color will happen after this time. So at this time, at this time, you will get SSW, smoke saturated water. Now this water is ready for giving the treatment to the plant, right? So you have to dilute it first, take 1 ml of this and 999 ml DDW. This will be a dilution of 1:1000, right? Then you have to take the second treatment, take 1 ml of this and take 499 ml of distilled water, dissolve it. This will be treatment 2. Similarly, for 250, you have to take 1 ml of this SSW and take 249 ml of DDW, dissolve it. This is treatment 3. The last treatment is 1:100. So take 1 ml of SSW and 99 ml of double distilled water. Check well. This will be treatment number 4. Now these chemicals are ready for spraying the plant. You can also study in hydroponics also, or tissue culture technique. You can use these concentration by different ways, right? So in this way, you can increase the speed. If the rate of generation of smoke is high, you can increase the speed. Okay? You can see the water is yellowing. Water is yellowing in color. So take a sample of 500 microliters after 5 minutes. Okay? And make a graph like this. Okay? When saturation occurs, there is no increase in optical density, stop. And now your water is ready, called as smoke saturated water, SSW, and you can just use for your treatment. On the other hand, you have to take a sample of this water, and also you have to perform the LCMS, okay, to find out that which fraction, how many fractions are there, and which fraction is KR1. Okay, if you are working on KR1, you can find out the KR1 in your piece of the LCMS, right? Clear to you? Any question, any query, any objection? This is the slowest speed, number 1. And now I am going to turn it off. Okay? Clear? Any question? Any suggestion? If you want to ask, you are welcome. I will be happy if you ask questions. Don't shy. What will happen? No more smoke will be dissolved in water if you increase the time, because the capacity of dissolving of this water to the smoke is saturated. Okay? This will be wasted. All the extra smoke will be sucked and ejected from this outlet. So it will be useless to run the system after maximum optical density is achieved. Okay? You have to take sample each after 5 minutes. Okay? Keep on taking optical density. When you find a saturation, stop it. Okay? Because no more smoke will be dissolved in this water, and this is fully saturated. Now you can use it after diluting it 1 to 1000 times, 500, 1 to 500, 1 to 250, 1 to 100, etc. Right? Clear? Thank you very much.
Thank you, Professor Masroor Saab. And very easily and very meticulously you have demonstrated how to develop plant-derived smoke-saturated water to apply on plants and generate and get its impact on plant. So in a very simple setup, he just demonstrated how to make such kind of water for use in increasing the yield and productivity of the plant. Thank you, sir, for your
Thank you.
informative demonstration.
scientifically known as an acetyl 5-methoxytryptamine. It's a biogenic amine that exists in almost all life forms, from microorganisms to plants and animals. It was the first discovery in 1858 in a pineal gland of vertebrates, where it plays a central role in regulating circadian rhythms, seasonal photoperiods, and immune responses. In plants, however, melatonin was only identified about 2 decades ago. Unlike in animals, where its role is well understood, the melatonin in plants has revealed surprising new function. One of melatonin's most important contributions in plants is its ability to enhance stress tolerance. It helps plants cope with various environmental challenges, including extreme temperatures, drought, salinity, and oxidative stress. In addition, melatonin regulates growth, promotes seed germination, delays senescence, and even play a role in photosynthesis. Melatonin has been identified in numerous plant families, including Poaceae, as grasses and cereals, Rosaceae, for example, fruits like apples and cherries, Apiaceae, carrots and parsley, Vitaceae is grape, and Brassicaceae, cabbages and mustard. So on this table presents melatonin in many plants, many species with common name, and of course the place the organ occurs. The right column indicates the concentration or level of melatonin in this plant. Interesting, the concentration of melatonin varies significantly dependent on the species, developmental stages, and environmental conditions. Some plants, such as rice and maize, have been found to certain particularly high melatonin level in the seeds, which suggest a crucial or important role in germination and easily seedlings development. Similarly, medical herbs have been reported to have a variable but often substantial amounts of melatonin, which may contribute to their health-promoting properties. Melatonin was the first found in phototropic marine organisms such as Dinoflagellata, Digulodium polyedrum, before it being detected in macroalgae and evenly in higher plants. In plants, melatonin is widely distributed across various tissue, including roots, leaves, flowers, seeds, and bulbs. However, melatonin level is uniform across species and even within different parts of the same plants. Several factors influence its concentration, including developmental stage of plants and environmental conditions. Of course, in algae there are also melatonin. For example, in Acutodesmus obliquus, my study indicate this fact. Another study has shown that seeds of maize and rice exhibit some of the highest melatonin concentration, ranging from 11 to 2,000 nanogram per dry weight in maize, and less in rice. On the other hand, we know that medicine plants have the similar level of melatonin. The high gas concentration was recorded in pistachio kernels, reaching between 227 234 microgram per gram dry weight. We know that this concentration is not static. For example, Arabidopsis thaliana leaves. In Arabidopsis thaliana leaves, melatonin levels range between 80 and 120 nanogram per gram dry weight. But these leaves do not fluctuate significantly through the day. Melatonin production is highly responsive, of course, to environmental stresses. Why melatonin biosynthesis in animals has been well understood for decades. Its pathway in plants is more complete and involves multiple subcellular localization. Melatonin biosynthesis begins with tryptophan, an essential aromatic amino acid. These molecules undergoes a series of enzymatic reactions to eventually form melatonin. The pathway consists of 4 key steps, each catalyzed by specific enzymes. You will see this name of these enzymes. Tryptophan is converted into tryptamine by action of tryptophan decarboxylase. Then, after hydroxylation into serotonin by tryptamine 5-hydroxylase, serotonin is acylated into N-acyl serotonin by serotonin N-acetyl transferase. Finally, N-acetyl serotonin is methylated into melatonin by N-acetyl serotonin orthomethyl transferase or coffeic acid or methyl transferase. Biosynthesis, of course, takes place in different organelles within the plant cell. Under normal growth conditions, melatonin biosynthesis follows 2 primary pathways. The first is from tryptophan, tryptamine, serotonin, and acetyl serotonin to finally melatonin, and the second from tryptophan via 5-hydroxytryptophan, then serotonin, N-acetyl serotonin, and finally to melatonin. However, when plants experience environmental stress or an increase in cellular serotonin levels, an alternative pathway may be activated from tryptophan via tryptamine, serotonin, 5-methoxytryptamine to melatonin. Regulation of melatonin biosynthesis is tightly controlled by mentioned enzyme activity. The key rate limiting step in this process is conversion of tryptophan to tryptamine, which is catalyzed by tryptophan decarboxylase. When plants need to increase melatonin production, the activity of this enzyme is often enhanced. Of course, additionally, the enzyme serotonin N-acetyl transferase and acetyl serotonin orthomethyl transferase, responsible for final steps of melatonin production, show increased activity under stress conditions. For example, drugs, high salinity or extreme temperature, high and low temperatures. This ensures that plant can rapidly generate melatonin when needed for protection. Unlike many others, phytohormones, melatonin is synthesized in organelles. We know that key enzymes, tryptophan decarboxylase, N-acetylserotonin orthomethyltransferase, and caffeic acid orthomethyltransferase are located in the cytoplasm. This suggests that the early steps of biosynthesis of melatonin, such as the conversion of tryptophan to serotonin, take place in these organelles. The next enzyme, tryptamine 5-hydroxylase, is found only in endoplasmic reticulum, which indicate that the hydroxylation steps converting tryptamine into serotonin is located in this organelle. Then next, of course, the last serotonin N-acetyl transferase is found both the chloroplast and mitochondria. The presence of this enzyme, for example, in chloroplast, melatonin may help photosynthesis and protect against oxidative stress. In mitochondria, it may act as an antioxidant to maintain cellular respiration and energy production. In the cytoplasm and endoplasmic reticulum, melatonin is likely involved in signaling and stress responses. This complex subcellular localization also suggests that during plant evolution, melatonin biosynthesis diversified to fulfill multiple physiological functions. The catabolism of melatonin is not fully understood. However, some research suggests that melatonin can be degraded via two distinct pathways. The first is a non-enzymatic degradation. It occurs through reactions with reactive oxygen species and reactive oxygen-nitrogen species. Melatonin can be hydroxylated or deoxygenated by free radicals, leading to the formation of various oxidative metabolites. Another important factors contributing to melatonin breakdown is UV light exposure, which can trigger direct photodegradation, similar to what occurs in animals. And the next pathway is enzymatic degradation. In plants, enzymes catalyze metabolism hydroxylation, leading to the formation of hydroxylated derivatives such as 2-hydroxymelatonin, for example, 2-hydroxymelatonin. These hydroxylation reactions are thought to be similar to those found in animals and other eukaryotes, indicated a conserved function in melatonin metabolism across different life forms in different organisms, plants or animals. The primary melatonin levels. Oh, I think that cherry fruits is rich in melatonin. The content is high in diffuse mulberry leaves and is higher in juvenile leaves than in older leaves. Oh, Yeah, higher levels of melatonin is often observed in seeds, wherein can elevate the damage caused by oxidative stress. Yeah. We know that melatonin production in plants is highly responsive to environmental stresses. Both high and low temperatures trigger melatonin synthesis as a defense mechanism. In Arabidopsis thaliana, cold stress significantly increase melatonin levels, helping protect plant from oxidative damage and chilling stress. Similarly, the heat stress in rice seedlings has been shown to enhance melatonin biosynthesis by activating key biosynthetic enzymes as a protective molecules, reducing water loss and maintaining cellular integrity and enhancing stress tolerance. Of course, melatonin does not act alone. Instead, it interacts with various classical phytohormones, forming a complex of signaling network that regulates plant growth, stress responses, and development. Melatonin and abscisic acid share overlapping functions, particularly in drought tolerance and stomatal regulation. Under drought stress, melatonin enhance abscisic acid biosynthesis, leading to stomatal closure, reducing water loss, and increased drought resistance in this plant. However, melatonin also acts as an ABA antagonist under normal conditions, preventing excessive abscisic acid accumulation that could hinder growth. Other phytohormones, auxin and melatonin, both influence root development and plant growth. Melatonin promotes lateral root formation similarly to auxin by activating auxin-related signaling pathways. On the other hand, in auxin-deficient plant, melatonin supplementation can partially compensate for auxin loss, demonstrating the functional overlap. Another phytohormones, cytokinins, well known for delaying leaf senescence, but melatonin also inhibited antisenescence properties, and in combination in cytokinins delay aging and improves chlorophyll retention. These interactions makes melatonin an important candidate for improving post-harvest shelf life in crops. Salicylic acid work with melatonin synergistically to enhance disease resistance, particularly against bacterial and fungal infections. When a plant is exposed to pathogen, melatonin can boost salicylic acid levels, activating immune response and reducing oxidative damage. In another interactions, melatonin can interact with jasmonic acid pathways to enhance wooding, healing, and secondary metabolite production, strengthening the plant defense mechanism. Understanding how melatonin interacts with these classical phytohormones open new possibilities in agricultural and crop improvement. Of course, by manipulation melatonin levels, we can improve plant stress resistance, enhance crop productivity and post-harvest quality, and the last develop plants can better withstand extreme environmental conditions. How melatonin regulates key physiological processes such as flowering, fruit ripening, and photosynthesis. Melatonin, of course, is a multifunctional molecules that regulates various biological processes. One of the best known function of melatonin is its strong antioxidant capacity. It neutralizes harmful reactive oxygen species, preventing oxidative stress that can damage cellular components such as DNA, protein, and lipids. We know after lectures, many asked for us said about oxygen stress and the catalase, peroxidase, and other antioxidant, non-antioxidant. Melatonin plays an active role in flower induction and reproductive development. It helps regulate photoperiodic resistance, ensuring that flowering occurs at the optimal time. It's good for agriculture, of course. In fruit-bearing plants, melatonin is involved in ripening processes, particularly through its interaction of ethylene and auxin. For example, in tomatoes, melatonin was found to promote ethylene biosynthesis, accelerating fruit ripening while also enhancing fruit quality and antioxidant properties. Melatonin also improved photosynthetic efficiency by protecting chloroplast from oxidative damage. Melatonin delays chlorophyll degradation, maintaining the higher photosynthetic rate and improving plant productivity. In crops such as wheat or rice, melatonin treatment has shown to increase biomass accumulation and enhance overall yield. Melatonin plays, oh, maybe other, oh, it's nice. At cellular level, melatonin protects chloroplast and reduces the red chlorophyll we know. Increased stomatal conducts, optimizing gas exchange and water regulation. Why is it important? Understanding how melatonin regulates fruit ripening, stress tolerance, and photosynthesis can we lead to practical application in post-harvest technology and crop management. By applying melatonin treatment, we can extend the fruit shelf life and quality after harvest, enhance fruit sweetness and weight, improving market value, boost photosynthetic performance, leading to healthier plants and increase crop yields. Melatonin also participates in regulation biotic and abiotic stresses. The combination of hydroxyperoxide and nitric oxide, mitogen activated protein kinase cascade, and oxidative signal inducible kinases are responsible for melatonin triggered innate immunity signal transduction. During this process, the gene encoding the salicylic acid biosynthesis enzyme isohorizmate synthase is upregulating, leading to increased salicylic acid levels. In summary, the protective properties of melatonin are: melatonin acts as a free radical scavenger, enhances activity accumulation of antioxidant enzymes, enhances the redox status of cell via regulation of glutathione synthesis and oxidation, ensures the protection of antioxidant enzyme from oxidative destruction. Melatonin reduces free radical formation, electron leakage by enhancing the efficiency of the mitochondrial electron transport chain. This graph summarizes the function of melatonin in plants. I hope we have a printed version. You may stay longer. The next group is strigolactones, a class of phytohormones that play, as I said earlier, a key role in plant communication, development, and stress adaptation. Unlike traditional phytohormones, strigolactones serve two major functions. The first, they regulate plant growth and architectures, much like auxin and cytokinins. But what makes them truly unique in the second role? The second role is chemical signals in soil, mediating interactions between plant and microbes. Strigolactones are carotenoid derivatives compounds naturally occurring in a wide variety of plants. Of course, not in algae. Not in algae. They were originally identified as root exudates, meaning they are released from plant root and into the soil. Strigolactones were first identified in 1966 when researchers studying cotton plants, similar, of course, to India, found certain compounds in the roots exudate triggered the germination of parasitic plants like Striga and Oronbache. Initially, these compounds were considered mere chemicals, substance released by plants and that influence neighboring organisms. However, decades of research revealed that they are actually a new class of plant hormones with far-reaching effects on plant physiology and biochemistry. The first naturally occurring strigolactone to be identified was strigol, from which the entire group gets its name. Since then, the scientists have discovered over 45 different strigolactones in various plant species. Some of the most well-known include Oronbacher, sargolactone, solanacol, and sargomol. Strigolactones are carotenoid derived lactones composed of three primary components. The first is ABA 3 cyclic core, a fuse ring system responsible for biological activity. And the next, the butanolide D ring, is has a crucial functional group that interacts with strigolactone receptors. And then connecting a known bridge, the structure likes the ABC core to the D ring and contributes to chemical stability. The D ring is particularly important because it serves as a bioactive signaling unit in plants and the rhizosphere. Some chemical structures, of course, strigolactone divided into canonical and non-canonical strigolactone, strigol, Oronbacher, heliolactone, zalolactone, avenol. More compounds are in the last book, Professor Shamsul, Professor Bahri, and me. edited, of course, not ours, book edited. We'll see on the first day. Strigolactons can be categorized into two main groups based on the structural differences. The first is canonical strigolactons. These compounds have a characteristic tricycling lacton, ABC structure connected to the butanolide D ring. Example includes on these slides. Canonically, strigolactons are primarily involved in shoot branching inhibition and mycorrhizal signaling. Non-canonical is the second type of strigolactons. These molecules lack the typical tricyclic ABC core, but still contain the bioactive D ring. They include simplified and modified derivatives from found in specific plant species. Non-canonical strigolactons are more diverse and often play a specific role in rhizosphere interactions rather than shoot regulation. Those molecules like the typical, we know about it. There's some examples are present. In 2008, strigolactons were categorized for the first time as a new class of plant hormones in recognition of their various role in controlling above ground plant architectures, for example, by inhibiting bud outgrowth and in underground communication with adjacent organism. Strigolactons were originally considered to belong to the sesquiterpene lactons, a widespread family of compounds formed in a cytosolic compound farnesyl diphosphate. Biosynthesis begins in the plastid, where carotenoids are broken down by carotenoid cleavage dioxygenases. The key enzyme in this process is dwarf 27. In abbreviation is D27, which convert all trans beta carotene into 9,6 beta carotene. It is followed by two critical cleavage reactions catalyzed by CCD7 and CCD8, resulting in formation of carotenoid-derived precursors, which are further modified to produce active strigolacton. The enzyme more axillary growth, as abbreviated as MAX1, cytochrome P450 monoxygenase, more known as MAX catalyze, a sequence of oxidation reactions converting carlacton, the first precursors of strigolactons, into carlactonic acid, and then is processed into methyl carlactonate, precursors the various strigolactons. Strigolactons serve dual function as both endogenous and exogenous signaling molecules. With the rhizosphere, these compounds are responsible for arranging the associated between plant roots and arbuscular mycorrhizal fungi, particularly in nutrient-deficient environment. They also play an important role in germination of parasitic plants, Striga and Orobanche. Strigolactons also plays other important roles in the root system, enhancing the length of primary roots and root hairs, encouraging growth and rise crown roots, and suppressing the creation of adventitious roots in, for example, tomato, Solanum lycopersicum, Arabidopsis thaliana, and Pisum sativum. Strigolacton play a dual role in underground signaling, as I said earlier, influencing both parasitic seed germination and the growth of beneficial mycorrhizal fungi. Their function in the rhizosphere determines whether a plant will form a mutualistic relationship or face a parasitic threat. In the left side, in the left side, when a host plant exudes environmental into the soil, these compounds act as a germination signal for dormant parasitic plant seed. In Striga or on Bache. Once activated, the parasitic seed germinates and begin searching for a host root. If the seed radical successfully reaches the host root, it established a parasitic interaction attaching itself to the host vascular system. It leads to nutrient depletion in the host plants, reducing its growth, weakening its structure, and in serve causes, causing crop failure. This interaction is especially problematic in cereal crops such as maize, sorghum, and rice, where parasitic weeds cause significant yield losses worldwide. Right side of this pictures present that strigolactons play a positive role in plant health by mediating beneficial symbiotic relationships with arbuscular mycorrhizal fungi. When a plant releases strigolacton into the soil, they are sensed by fungal hyphae, which respond by directing their growth toward the plant's root system. Once contact is established, the plant and fungus exchange essential nutrients. The plant provides sugars and as an energy source to the fungi. In return, the fungi supply nitrogen, phosphorus, or other nutrients to the plant, enhancing growth and stress resilience. This mutualistic relationship is particularly important in nutrient-poor soil, as it significantly improves plant nutrient uptake and overall fitness. Strigolacton influences key development and traits, including seed germination, early seedlings development, ensuring optimal establishment, internode elongation, affecting plant height and canopy structure, leaf morphology and senescence, helping plants adapt to environmental conditions, should gravitropism, enabling plants to adjust their growth direction in response to gravity, stem morphology, influencing plant architecture and biomass distribution. One of the most striking effect of strigolactons is their role in accelerating leaf senescence. Strigolacton interact with abscisic acid, another key phytohormone involved in stress signaling and draw response. We know that strigolacton play an important role how plants respond to osmotic stresses and drought conditions. Of course, they interact with abscisic acid, regulate root-to-root signaling, they influence stomatal closure, all of which contribute to water conservation and stress adaptation. In normal conditions, strigolacton are produced in root and transported upward to the shoots where they suppress the transcription of strigolacton biosynthesis genes, ensuring that strigolacton levels remain balanced. However, during drought or osmotic stresses, the synthesis of strigolacton in roots is inhibited, reducing the upward flow of strigolacton molecules. As a result, strigolacton biosynthesis genes in shoot became active, more active again, leading to an increase in strigolacton production within a shoot itself. Once produced in a shoot, strigolacton contribute to stomatal regulation, helping plants minimize water loss. Of course, it's summarizing. It means that strigolacton act as a both direct and indirect regulators of stomatal movement, ensuring that plants retain water efficiency during stress conditions. Strigolactons also plays important role in plant immunity. These hormones influence gene expression, interact with key defense-related hormones, and even inhibit pathogen growth, making them important regulators of plant resistance against bacterial fungal infections. Application of synthetic strigolactons was found to strongly inhibit the formation and growth of phytopathogenic fungi. So strigolacton not only modulate plant immune responses, but also act directly against fungal pathogens, potentially offering new strategies for natural plant protection. Strigolacton may also help plant overcome pathogen-induced stress at the transcription level. They interact with the proteinase inhibitor 2, PIN2 gene, which plays a role in plant wound and pathogen response. Of course, strigolactons contribute to a fine-tuned immune response, ensuring that plant efficiency defends itself without unnecessary energy expenditure. Strigolactons act as a mobile signaling molecules that are synthesized mainly in roots and transported to the target tissues, where they influence various physiological processes. In this target cells, strigolactons are perceived by specific receptor protein called dwarf called dwarf 14. This receptor belongs to the alpha-beta hydroxylase superfamily and plays a dual role. it not only bind strigolacton but also hydrolyze them, triggering the signaling cascade. Why once strigolactons are detected by D14, the receptor interacts with MAX2, we know the full name, and F box protein that plays a key role in signaling transduction. MAX2 is a part of ubiquin ligase complex which function as a molecular switch that regulates plant growth responses. Under normal conditions, a receptor protein known as dwarf 53 inhibits the downstream effects of strigolacton, preventing changes in gene expression. However, when strigolactons are present, D14 MAX2 complex mark D53 for degradation, effectively removing its inhibitory effect. D53 is degraded, genes responsible for stractolacton response became active. It leads to several physiological changes such as the inhibition of shoot branching, modification of root growth pattern, and improved adaptation to environmental stresses. In a sense, the degradation of D53 allows strigolacton to exert the full effect, ensuring thus the plants can adjust the growth and development based on external or internal cues. Strigolacton and maybe I told interactions. Yeah, it's probably yes. It's the most probably. This is the end. Thank you. Tomorrow I will have the last lecture, introduction to phytohormones, steroidal regulators, similar to brassinosteroids. They are not typical brassinosteroids, but occur in animals, insects, and plants. Thank you.
Thank you, Professor, for a hectic schedule you have, and so many lectures now you have completed, and still one lecture is about the new plant hormone and the editosteroids is left. That will be tomorrow, and that will be the last lecture for him. Okay? Yeah.
Phytohormones are not phytohormones,
Yeah.
but plant growth
Regulator. Yeah. That is a. But in
11 typical phytohormones plus peptide hormone.
But in my opinion, in the coming year, it might be accepted as a hormone, plant hormone. Because it has most of the properties of the phytohormone. Yeah. So that's why we are thinking that we will get a new plant hormone. No? Yeah.
When you get my presentation,
Yeah.
will be very good.
Yeah. That's why I'm telling you. Yeah. Because we have a common review on the phytohormones, so we know that what the phytohormone is. Yeah, okay. tissues or organs under a sterile condition. That is the one point. And another is that it is basically used for clonal propagation, genetic modification, and also produce a disease-free plant and the germplant conservation. We have a different type of the tissue culture, that is organ culture, meristem culture, protoplast culture, as well as that of the cell culture. This is the one of the example of the organ culture. Overall procedure are there, that what are the different steps are going to be taken place, and this the steps for the selection of the explant is basically same in all of them. So Mr. Sawahat will tell you in the laboratory condition. That's where I am not going to give in the detail itself. Then we have another type of the culture that is called the meristem, where we are just removed the meristem from the older leaf and then form the calyx, and finally the plantlet will be formed. It is just a we have just draw a diagram of this, so you will see everything in that of the laboratory condition. Then we have a protoplast culture, that is one of the important. Basically the protoplasts are cell without a cell wall, and but bounded by a cell membrane or the plasma membrane. So using the protoplast, it is possible to regenerate whole plant from a single cell and also develop a somatic hybrids. So these are the different various steps which has been required for the culture of the protoplast. Then we have another type that is known as the cell suspension culture. So basically the growth of the cells, including the culture of single cells or small aggregates of cells in vitro in a liquid medium, is known as a cell suspension culture, and it is helpful in the production of the secondary metabolites. These are some of the secondary metabolites which has been isolated from the different plant sources, such as the digoxin. One of them is found in the digitalis purpurea, and it has been helpful in the cardiac tonic, and so on. These are the list of the different secondary metabolites which had been isolated from the different plant sources. Then what are the key steps in tissue culture? These key steps are basically told by the Sawahat, that first we have to explant selection. Whatever we are going to select, that will be a perfect one. Generally a small part of the plant are used, that is known as a explant. Then we have to sterilize. So to prevent the contamination from the microorganism, we have to sterilize our instrument, our plates and everything that has been used to inoculate the explant. Then we have the culture media preparation, so a nutrient solution that support the growth and differentiation that will be made. Then the first step that is going to be formed is the formation of the calyx from the explant. That is, it is basically an unorganized cell growth, often induced by the hormone. So out of the 6 major hormones, the auxin play an important role in inducing the calyx formation. So that's why we are telling that the tissue culture as well as that of the plant hormone have a direct link between each other. Then we have the organogenesis, that is from the calyx itself the root and shoots are formed. So that process is basically come under the category of the organogenesis. These are the various steps which will be used, that will be told by our students. Then the same thing is detailed, that is small tissue we have to select healthy disease-free plant. Then size and age of the explant, as well as that must be free of the contamination. Then these are the what are the way that has been included for the selection of the explant. Then these are the various steps or various things which has been, this is your laminar flow where most of the inoculation has been done. So this should be sterilized before doing, and all the things which has been just kept it here, these are sterilized in the your autoclave. Okay, so after sterilization, everything has been put, and this is the that how it is going to be explanted. So these are the various steps which has been included. Then we have made a culture media generally for the growing the callus as well as the rudder. One of the major media that has been used is known as the MS media and Murashi's Skog media. Here it is the constituent that is going to be there. This then we have the first step is that the chylus formation, and the finally then it is a organogenesis. These are the that chylus I have already told you, and chylus can be differentiated into shoot and root. The same thing is there. So, but as if you remember my class in the MSc second semester, where I was teaching the auxin as well as that of the cytokinin, I always told you that if there is a proper balance between auxin and cytokinin, and that balance basically create the formation of the root and shoot. The same thing is here, that the hormonal balance between the auxin and cytokinin is required for the formation of the root and that of the shoot. Then this is the complete process. Then what is the basically the how you can relate the tissue culture into that of the plant hormone itself. These are the different plant hormone which has been basically used, that is the gibberellin, auxin, cytokinin, ethylene, abscisic acid as well as that of the cytokinin. These are the 5 different hormones which are basically function for the plant hormone in the tissue culture. This function gibberellin, shoot and root, apical meristem. Then we have that of the auxin, basically the apical meristem, as well as that of the immature plant tissue, and so on. We have a different function. So it is here. It is not a theory class. It is just a demonstration for the tutorial. So, but everything is written here. You can go through it. It is not possible to go through each and every point at every time. Then you have the auxin, how it is going to form the embryogenesis, how you are going to form the root, and then how it is going to form the shoot. So these are the different concentration, auxin and pin. That is your cytokinin is denoted here as a pin. Then we have here cytokinetin. One type of kinetin is basically used, and here you will get this type of the structure, that is their chylus. Then we have also the role in the gibberellin. Basically, it promotes the cell elongation, germination, and flowering. Just here, it is the some of the picture which has been obtained in the one of the experiment conducted in the tissue culture laboratory, that where they have used the different concentration and then got a such type of the result. Then we have a role of abscisic acid. Basically, it involved in the stress response, seed dormancy, as well as that of the abscission process. These are the some of the plant species where the role of these are the ABA long list, and these are the references. And so it is too much. It is not point to. It is not going to be just read one by one. There is no need. And then we have the formation of the ethylene. That gaseous hormone, it is basically involved in the fruit ripening and flowering as well as that of the senescence. Just see this structure, and here if you will just, picture is not clear here, if you just elaborate it in your mobile, then you can get which concentration is going to just affect the flower formation as well as that of the color. But then what is the application of the tissue culture technology? First is that it is going to use for the colonal propagation of the plant, that is called as the micropropagation. Then we have the somatic embryogenesis, that is the production of the embryos from the somatic cells. Then third one is the gene editing, or that of the transformation, that inserting a particular gene into a particular plant that will not, you can say, inhibit the growth or any other thing. Then also we are going to produce a pathogen-free plant from this application. This is a just various step, a direct or indirect step for the somatic embryogenesis, then we have a de novo organogenesis, where other two steps, that is the direct and indirect steps are there. Then these are some example of the that how we are going to insert the gene, and then we have got a some of the change that you have a golden rice, flowers of tomato, BT corn, eco advantage. These are the some of the plant which are basically after incorporating the gene, and this, that has been produced through the tissue culture. They are the normal and the GMO crop. But you just see there is some example here. If that a particular gene is there, and here that is called the GMO crops. Just see that the difference between the GMO crop as well as that of the normal crop. GMO crop is basically a disease-free crop, and the normal crop have a different type of the diseases are there. So finally, we led to a conclusion that tissue culture is basically is a powerful tool in plant biotechnology, and the plant hormone, without plant hormone, the tissue culture cannot be done, because plant hormone are essential for regulating plant growth and development in the tissue culture. But there is a combination of various hormone, and the combination of various hormone determine the success of tissue culture technique. These are the few important points which has been basically used in relation to the tissue culture. So it is all about the basic important point things. Now 10 by 10 student can move there to the into tissue culture laboratory, and where they will tell you how to inoculation has been done. Okay. Ms. Samin Zaidi, please take them 10 students to there.